Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 2, Host Vector System. A successful biotechnology experiment depends not only on the DNA that we want to use, but also on the biological partnership between the vector that carries it and the host cell that must receive, maintain, and use it. By the end of this lesson, you will be able to choose a suitable host vector pair, explain why it works, and identify the evidence needed to prove the intended result. Keep your notebook ready, pause whenever you need to study a diagram, and let us build the chapter step by step. Here is everything we are going to learn. We will first understand host vector compatibility through entry, replication or maintenance, selection and intended output. We will examine why Escherichia coli is widely used, where it has limitations, and when yeast, plant or animal cells are better hosts. We will study the essential anatomy of a cloning vector, including the origin of replication, selectable marker and cloning site. We will then compare plasmids, bacteriophage-derived vectors, cosmids, bacterial artificial chromosomes, and yeast artificial chromosomes. Next, we will learn how to select a system from the required outcome and insert size. We will follow DNA delivery through disarmed TI plasmid systems in plants and examine vector strategies for animal cells. We will distinguish cloning vectors from expression vectors, understand shuttle vectors, and connect promoter, reading frame and regulatory signals to protein production. Finally, we will use controls and an evidence ladder to verify DNA, expression and function, and we will practice structuring clear, full-mark CBSE answers. With that roadmap in mind, let us begin with the central question of the chapter. Imagine placing exactly the same gene into three cells: a bacterium, a yeast cell, and a human cell. The DNA sequence has not changed, yet the result may be completely different. One cell may copy the DNA efficiently, another may produce a protein, but modify it incorrectly. A third may fail to recognize the vector at all. This is the central idea of the host vector system. A vector is not simply a DNA container, and a host is not simply a living test tube. They must function as one engineered biological system. In this lesson, we will learn how to choose that system, how to read a vector map, how plasmid, phage, plant and animal vectors differ, and how to prove that the chosen pair actually works. Keep one question in mind. What evidence would show that a host vector pair is suitable for the purpose, rather than merely present in the same experiment? Host vector compatibility means that the vector can enter the chosen host, be recognized and maintained there, and support the intended experimental outcome. Think of 4 gates. Gate 1 is entry. Can the delivery method move the vector across the cell's barriers? Gate 2 is maintenance. Does the host recognize the vector's origin of replication, or can the DNA integrate or otherwise persist? Gate 3 is recovery. Does the selectable marker function under the chosen conditions, allowing vector-containing cells to be enriched? Gate 4 is outcome. If expression is required, can the host recognize the promoter, translate the message, and process the product correctly? Passing one gate does not guarantee the next. Antibiotic-resistant colonies show that selection worked, but do not prove the insert, its orientation, or a functional protein. A full explanation therefore connects vector feature, host function, observed result, and validation test. Escherichia coli is a widely used host because it grows rapidly, is comparatively inexpensive to cultivate, is genetically well characterized, and accepts many engineered plasmid and bacteriophage systems. Laboratory strains can be selected for useful properties such as improved DNA uptake, reduced unwanted recombination, reduced nuclease activity, or compatibility with a particular expression system. These advantages make E. coli excellent for routine DNA propagation and for producing many proteins. But rapid growth is not the only criterion. Bacteria do not perform every eukaryotic RNA processing step or post-translational modification. A eukaryotic genomic gene containing introns may not be expressed correctly, and a protein requiring complex folding, glycosylation or secretion may not become biologically equivalent to its native form. The exam quality conclusion is balanced. E. coli is efficient when its molecular capabilities match the product, but unsuitable when biological fidelity requires machinery it does not possess. Eukaryotic hosts are chosen when the biological outcome requires cellular machinery that bacteria cannot provide. Yeast combines relatively convenient cultivation with some eukaryotic processing and is useful for many recombinant products. Plant cells are appropriate when the goal is a heritable plant trait, plant-specific expression, or regeneration of a complete transformed plant. Animal cells can support complex folding, secretion and modifications needed by many animal proteins, although culture is generally slower, more demanding and more expensive. Host choice is therefore a trade-off among fidelity, yield, speed, cost, stability and biosafety. Consider 3 products. A simple cytosolic enzyme may be suited to bacteria. A glycoprotein whose activity depends on processing may require a eukaryotic system. A stable crop trait requires delivery to plant cells, followed by regeneration and inheritance testing. The best host is not the most sophisticated host. It is the simplest system that reliably produces the required biological result. Open a generic cloning vector map. The origin of replication answers where and under what host machinery the vector can replicate. It also influences copy number, which affects DNA yield and sometimes cell burden. A selectable marker answers how cells carrying a relevant vector-associated property can be enriched. A cloning region or multiple cloning site provides defined positions for inserting DNA. Beyond these basics, size, structural stability, host range and compatibility with other replicons affect performance. An expression vector needs additional signals: a host-compatible promoter, translation initiation information, a transcription terminator, and sometimes regulatory, purification, or secretion elements. Each label must be connected to function. The origin does not select colonies, the marker does not prove insert identity. The promoter does not guarantee a correctly folded protein. In a CBSE diagram, draw a clear circular or linear map, label only relevant elements, and explain beside it how each feature supports replication, selection, cloning, or expression. Plasmids are usually circular, independently replicating DNA molecules that have been engineered into highly practical cloning vectors. When you interpret a plasmid map, do not merely identify the origin, marker, and cloning site. Turn the map into an experiment. Ask whether the chosen restriction sites cut the vector once, whether they preserve required elements, and whether using 2 different ends can control insert orientation. After ligation and transformation, selection identifies cells with the chosen vector-associated marker. Screening then tests likely recombinants, and restriction analysis (PCR) or sequencing confirms the final construct. Copy number also matters. A high copy plasmid can provide abundant DNA, but excessive copy number or expression can burden the host or destabilize a difficult insert. 2 plasmids may also be incompatible if their maintenance systems interfere. The general lesson is that a plasmid map predicts possibilities. Experimental evidence establishes what molecule was actually recovered. No single vector class is best for every DNA fragment. Standard plasmids are convenient for many routine inserts, but larger fragments require systems designed for greater capacity and stability. Bacteriophage-derived vectors exploit efficient infection or packaging steps. Cosmids combine plasmid-like maintenance with phage packaging signals. Bacterial artificial chromosomes maintain large DNA fragments in bacteria, often at controlled copy number for stability. Yeast artificial chromosomes use yeast chromosome-like elements to maintain still larger DNA segments in a eukaryotic host. The exact choice depends not only on size but also on library purpose, stability, host, recovery method, and downstream analysis. A genomic library requiring broad coverage may benefit from large inserts because fewer independent clones are needed, but larger constructs can be harder to manipulate. For an exam comparison, arrange the vector classes by general capacity, then add the biological maintenance principle and the experimental purpose. Names alone do not explain selection. Let us solve a host vector choice rather than memorize one. Suppose the goal is to propagate a modest DNA fragment for sequence analysis. A compatible bacterial plasmid and E. coli may provide speed, easy selection, and abundant DNA. Now change the goal to production of a protein requiring a particular eukaryotic modification. The same plasmid-host pair may copy the gene yet produce an unsuitable product, so a compatible eukaryotic expression system becomes more appropriate. Change the goal again to construction of a genomic library containing very large fragments. Vector capacity and stability now dominate, pointing toward an artificial chromosome system rather than a routine plasmid. Notice the reasoning order. Define outcome, estimate insert requirements, identify host biology, select vector features, plan delivery and selection, and finally choose confirmation tests. If two systems could work, compare yield, fidelity, stability, cost, and safety. This decision chain is far stronger than answering with a vector name alone. Agrobacterium naturally transfers a defined DNA region from its TI plasmid into plant cells. Biotechnology adapts this ability by disarming the disease-causing components while retaining the transfer logic. The DNA intended for the plant is placed between transferred DNA border sequences. In a binary vector strategy, the engineered tDNA region and the bacterial functions that support transfer can be organized on compatible molecules. Agrobacterium contacts plant tissue, the transfer machinery processes the border DNA, and the engineered sequence reaches plant cells. But a transformed cell is not yet a complete validated crop. Selected cells must be regenerated into plants, the insert must be confirmed molecularly, expression and phenotype must be tested, and stable inheritance may need to be demonstrated. In a labeled flowchart, show engineered tDNA, bacterial delivery, plant cell entry, selection, regeneration and verification. Avoid saying that the entire tumor-inducing plasmid is simply inserted unchanged into the plant. Animal cell vector design begins with a different set of barriers. The DNA must enter the cell, often reach the nucleus, persist for the required time and use regulatory signals recognized by the animal cell. Plasmid-based delivery may be suitable for transient expression, where the product is needed for a limited period. Viral-derived systems can provide efficient entry and, depending on the design, longer persistence or integration. These advantages introduce additional questions. What is the host range? How much cargo can be carried? Is integration intended? Could insertion disrupt cellular DNA? And could viral components trigger an immune response? Production purity and replication safety also matter. Do not classify one strategy as universally superior. Match the vector to the cell type, duration and acceptable risk. Then distinguish evidence. Reporter expression can show delivery, molecular analysis can test persistence or integration, and functional and safety assays test the biological consequence. An expression vector is designed not merely to maintain DNA, but to produce RNA or protein. The promoter must be recognized by the chosen host and may be constitutive or regulated. Translation initiation signals must work in that host, the coding sequence must remain in the correct reading frame, and transcription termination must be reliable. Tags may help detection or purification, while secretion signals may direct the product to a useful cellular location. Yet every added element introduces a condition to verify. Strong expression can burden or harm the host. A protein can be abundant but insoluble, incorrectly processed or inactive. Use an evidence ladder. DNA analysis confirms the construct. RNA analysis tests transcription. Protein detection tests production and approximate size. Purification data assess recovery, and an activity assay tests function. For a full credit answer, label the expression cassette in order and connect each element to the molecular event it controls. A shuttle vector is engineered to operate in two different host systems. A common workflow constructs and amplifies the recombinant molecule in bacteria, where DNA manipulation is convenient, and then transfers it to yeast or another eukaryotic host for analysis or expression. To do this, the vector carries host-specific functions for both environments. It may contain a bacterial origin and bacterial selectable marker together with a yeast replication element and yeast selection marker. These components do not become universally active merely because they share one DNA molecule. Each functions only where the host can recognize it. The vector must therefore be tested independently in both hosts. A bacterial colony proves bacterial maintenance, not yeast expression. In an exam diagram, divide the map into bacterial and eukaryotic modules, then draw the workflow from construction to amplification to transfer to selection and outcome in the second host. How do we prove that a host vector system works? Begin by matching evidence to the claim. Growth under selection suggests that cells possess the selected property, but an untransformed negative control is needed to show that the condition is selective. An empty vector control separates effects of the vector and host from effects of the insert. PCR, restriction analysis or sequencing, can test insert presence and structure. If expression is claimed, compare RNA or protein with suitable controls. If biological function is claimed, use an activity or phenotype assay and include a positive comparison where possible. Replicates address reproducibility, while an independent method reduces the chance that one technique created a false conclusion. State the direct observation before the inference. For example, a band of expected size is an observation. Correct insert identity is an interpretation that may still require sequencing. Good biotechnology moves from observation to supported inference to a clearly limited conclusion. For a 3 or 5 mark answer, start by defining the host vector system as a compatible combination that supports entry, maintenance, recovery, and the intended cloning or expression outcome. If asked about vector features, draw a large map and label the origin, selectable marker, and cloning region. Add promoter and expression signals only when relevant, and state the function of every label. For a comparison, keep the bases parallel: host, capacity, maintenance, delivery, expression, and use. For a plant vector answer, write the sequence from engineered tDNA to delivery, selection, regeneration, and molecular conformation. For an expression vector, preserve the direction from promoter through coding sequence to termination. Avoid 3 common errors: choosing a host only because it grows quickly, treating a selectable marker as proof of the insert, and listing vector names without connecting them to size, host, and purpose. Finish with the validation required for the claim. Close your notes and rebuild the chapter as a decision chain. Begin with the intended outcome: DNA propagation, a library, a protein, or a stable trait. Define insert size and biological requirements. Choose a host with the necessary replication, expression, and processing machinery. Choose a vector with compatible maintenance, selection, cloning, and, when needed, expression elements. Plan delivery and recovery. Confirm the construct, then test expression and function at separate evidence levels. Now answer 4 questions. Why can a plasmid replicate in one host but fail in another? Why might E. coli produce the wrong form of a eukaryotic protein? Which features must a bacteria yeast shuttle vector carry? And why does a selected colony not prove the correct insert? If your answers link mechanism to evidence, you understand the host vector system. The chapter is not a catalog. It is a framework for engineering a biological partnership and proving that it performs the intended job.